Hello everyone. So, in this section I am going to discuss about the acid regulation and the formation and role of ATP, then creatine phosphate and basal metabolic rate. So, we will start with the acid regulation, what is the gastric juice, how the acid is regulated, and then we will cover all of the other points, the ATP and creatine phosphate and BMR. So, let us start with the gastric juice. Now, about 2 liters of the gastric juice is secreted daily by the specialized secretory glands, yes, and these secretory glands are present in the mucosa lining. Now, the gastric juice, which is we are saying here it has 2 liter, it is consisting of water and mineral salts, yes. Mucus is secreted by the mucus neck cells in the glands or the surface mucus cells present in the stomach surface area, yes. So, these all are under the gastric juice, means the water, mineral salts or the mucus. Now, hydrochloric acid is also one of the part in the gastric juice, the content or the juice present in the stomach. So, HCl is there, and this HCl is produced or secreted by the parietal cells. Similarly, intrinsic factors are there, it is also produced from the parietal cells present in the gastric glands. Now, the intrinsic factor is very useful for the absorption of vitamin B12, yes, and if there is a deficiency in the intrinsic factor, that can lead to the deficiency in vitamin B12 as well. Now, inactive enzyme precursors are there like pepsinogens. Now, these pepsinogens are produced by gastric chief cells in the gastric glands. The pepsinogen we are saying it as inactive because they are produced as an inactive form, but this pepsinogen converted into pepsin with the help of the hydrochloric acid present in the gastric juice, and that particular pepsin will ultimately useful for the metabolism of the proteins to the simpler form of peptides. Now, coming to the functions of the gastric juices, or so the first function here is the water which is present in the gastric juice is liquefying the food which we are swallowing, yes, whatever we are ingesting, the food we are ingesting, that will be properly liquefied with the help of water present in gastric juice. Then there is a hydrochloric acid present. That hydrochloric acid, the first thing it will do, it will acidify the food and stopping the function of the salivary amylase, which is coming from the salivary glands. Okay. So, first function the hydrochloric acid will do this, and then this hydrochloric acid is also beneficial for killing the ingested microbes, okay, whatever the microbes which is coming through the food, they will be killed off. Then it is also providing the acid environment which is required for effective digestion by pepsin. I already told you about how the pepsin are formed in the presence of hydrochloric acid. Now, coming to the pepsinogen which is present in the gastric juice. This pepsinogen is activated to form of pepsin, and they will ultimately required for the digestion of the proteins. Now, these enzymes, they will begin the digestion of proteins by breaking them into a smaller molecules, smaller form of peptides. Now, the pepsin which is the activated form of pepsinogen, it is mostly effective at the pH of 1.5 to 3.5, yes, means that is the acidic pH. Now, the intrinsic factor I told you, which is produced or secreted by the parietal cells, that is required for the absorption of vitamin B12 from the ileum region, from the intestinal region, yes. Now, we will see the mucus. So, mucus is produced in the stomach lining, and this mucus is very beneficial for prevention against the mechanical injury, a mechanical injury due to the HCl which is present in the stomach, yes. So, the mucus is preventing the mechanical injury to the stomach while by lubricating the content, first thing. Second, it prevents the chemical injury by acting as a barrier, yes, between the stomach wall or the corrosive gastric juice, means the hydrochloric acid. Now coming to the secretion of gastric juice, how it is secreted, yes. So, there is always a small quantity of gastric juice present in the stomach, even we are on the fasting. We are not taking any food, at that time also there is some or very small amount of gastric juice present, which is containing all of the agents I told you, like water, the mucus, yes, then there is a pepsinogen, hydrochloric acid, intrinsic factor, these all are present, yes, but in a very small quantity. Now, secretion reaches its maximum level about 1 hour after the meal. When we will take the meal, within the 1 hour, yes, there will be that the gastric juice, the secretion of this gastric juices, that will attain its maximum level, yes, and it will starts to decline, the secretion will starts to decline when or after, you can say after the 4 hours of the meal, yes. Now coming to the different phases. So, the secretion of the gastric juice, we have divided it into 3 phases. The first phase is the cephalic phase, the second phase is called the gastric phase, and the third phase is called the intestinal phase, yes. Now, we will see how they are classified, yes, what do you mean by cephalic or the gastric, yes, and the intestinal phase. So, in the first phase, the cephalic phase, this flow of juice in this particular phase, the flow of a juice occurs before food reaches the stomach, yes. So, this is important here, before reaching the food in the stomach, the flow is started, yes, and how the flow is started? See, it is due to the reflex stimulation of the vagus. There is an involvement of vagus nerves, yes, there is a stimulation of the vagus nerves, the parasympathetic nerves, yes. Now, this parasympathetic nerves, they are stimulated through the, we have seen the food, yes, through the sight, whatever we are seeing, yes, the smell of the food, yes, or the taste of the food, yes, whatever we have ingested and it is in the mouth just, and with that particular taste, or just with the reflex also, yes, whatever our past experiences with that particular food. So, that can also stimulate the vagus nerves, and that can stimulate the flow of the gastric juice, yes. So, this is known as a cephalic phase. Now, coming to the second part, that is a gastric phase, yes. After the cephalic phase, gastric phase will be started. Now, in this the gastric phase is stimulated by the presence of food, yes, and in this what happens, the enteroendocrine cells, they are present in the pylorus, yes, and duodenum area. From this pylorus and duodenum, there the enteroendocrine cells will secrete the gastrin, yes, which is a hormone, and that will pass directly into the circulating blood, and this particular, this gastrin is here regulating the gastric juice, yes, the flow of the gastric juice. Now, gastrin, which is reaching in the bloodstream, which is circulated in the blood, that supplies the stomach, that will stimulate the gastric glands, which will produce more of the gastric juice, yes. So, this is how the gastric juice is regulated in the gastric phase. This is only due to enteroendocrine cells, which is releasing the gastrin, and that particular gastrin is stimulating to produce more of the gastric juice, yes. Now, gastric secretion or gastrin secretion is suppressed when the pH in the pylorus falls to 1.5, yes, when there is the pH is nearly to 1.5, the secretion of the gastrin will be suppressed. Now see, the third phase is the intestinal phase. Yes. In the intestinal phase or the last phase, yes, the partially digested contents of the stomach, it will reach in the small intestine. Yes, this intestinal phase is started in the small intestine. What happens here is the hormones such as secretin or cholecystokinin. These hormones are produced by the endocrine cells in the small intestine. Yes, these are the endocrine cells present in the mucosal lining. Now, these hormones they will cause the production of the bile salts, yes, cause the contraction in the gallbladder, that will liberate the more of the bile juices, that will cause the emulsification of the fat. So, this is one of the function like secretin and cholecystokinin. Secretin is causing the production of the bile, whereas the cholecystokinin is causing the contraction of the gallbladder. Now, there is they slow down the secretion of gastric juice and reduce gastric motility. In this phase, what happens here is the secretion of the hormones, yes, the secretin and cholecystokinin. With these hormones, they are causing the slowing down of the secretion of gastric juice, yes, in the stomach area, and they are reducing the gastric motility, so that the content will not move in a backward direction. Yes. Now, the rate at which the stomach empties, that depends on the type of the food eaten. Yes. So, if a person had taken the carbohydrate meal, a carbohydrate meal leaves the stomach in 2 to 3 hours, whereas the protein will take longer time. Yes, and the fatty meal, that will take more time. Yes. So, this is depends on how the food is, what type of food you have taken. Now, this is the one diagram you can see, this is the one conclusive diagram in which you can see all of these 3 phases. So, let us understand this diagram here. First phase, there are 3 phases. So, first phase is the cephalic, the second phase is the gastric phase, and third phase is the intestinal phase. Cephalic phase start from the stimulation of parasympathetic system or stimulation of vagus nerves. They are stimulated through the sight, smell, taste, thought of the food. Yes, we are thinking about our favorite food, that start to produce the gastric acid. Yes. So, this is all the reflexes, they will work here. Now, the vagus nerves are stimulated due to the thinking of all of these. We have seen, we have the smell or a taste or the thought of the food, they will stimulate the vagus nerves, that will stimulate the secretion of gastric juice. Yes, this is up to the cephalic phase. Now, the food is reached in the stomach. When the food is reached in the stomach, the second phase starts, the gastric phase. In this, the food in the stomach stimulates secretion of the hormones. Which hormone is secreted here? Through the endocrine cells, that is the gastrin. Yes, the gastrin is stimulated, and that gastrin in the bloodstream, that will liberate the more of the gastric juices. Yes. So, this is how the gastric phase is regulated. Yes. Now, the food is moved to the small intestine, the partially digested food, because the completely it will digest in the small intestine. So, we are saying it is a partially digested from this stomach, that will move to the small intestine. Now, the small intestine, there is a one hormone, that is a enterogestrone. Yes, this particular enterogestrone is formed from this particular region, the lining in the small intestine, and this particular hormone, what it will cause? The food in the duodenum stimulates the secretion of the hormone. Once the food is reached in the small intestine area, then it will cause the stimulation of the secretion of the enterogestrone, and that particular enterogestrone will cause the reduction of the secretion of gastric juices. So, here it is written enterogestrone in the blood reduces gastric motility and secretion of gastric juices. This is how it is regulated. Yes, it starts from the cephalic with the thinking of the food and end up to when the food is reached to the small intestine, which is called intestinal phase with the help of enterogestrone. Yes. So, this is the whole mechanism. I hope this is clear to all of you about the phases of gastric secretion. Now, coming to the another topic, the formation and role of ATP. Yes. So, ATP, adenosine triphosphate. This is the energy source, main energy source. Yes. For most cellular process, if you will see the metabolic pathways, central metabolic pathways, which is followed for the metabolism of carbohydrate, proteins or fats, like glycolysis is there, then TCA, tricarboxylic acid cycle or Krebs cycle, yes, it is there. Then we have oxidative phosphorylation or electron transport chain reaction is there. In all of these processes, we are seeing the ATP is generated. This is a very basic thing, or cell is surviving just because of this ATP, this energy. Now, the building blocks of ATP, if we will see the structure of the ATP, adenosine triphosphate, there is the involvement of carbon, nitrogen, hydrogen, oxygen and phosphate, the phosphorus. Yes. Because of the presence of unstable or high energy bonds present in adenosine triphosphate or ATP, it is easily hydrolyzed. Yes, it is easily hydrolyzed in reactions to release large amount of energy. Yes. Now, you see here in this diagram, when energy is not needed by organism, yes, at that time the phosphate group is added back to AMP, adenosine monophosphate, yes, or and ADP, adenosine diphosphate, to form ATP. When energy is not required, the phosphate will come with the AMP or ADP and that will form the ATP, that is a stored form. Yes. Now, this can be hydrolyzed later as per required. Whenever the energy is required, the ATP will further break down into AMP or ADP, it will release its phosphate. So, you can see this diagram here. Now, this ATP is hydrolyzed and it will liberate the ADP, adenosine diphosphate, it has liberated only one phosphate. If it will liberate the second phosphate, it will cause AMP, adenosine monophosphate, and another phosphate group will be liberated from this. So, in this whole process, when the ATP is formed, the ADP, adenosine diphosphate, there is the energy formation. So, energy you can see here, energy for cellular work, yes, endergonic or energy consuming processes. Here we can see the cycle is repeated here, the ADP that can be attached with the phosphate when the energy is not required by the organism and can be form the ATP again. So, energy from catabolism or exergonic energy releasing processes. So, here it can also form the ATP, it can also liberate the energy in the further the when there is a conversion to ADP. So, this is how the ADP or ATP is formed. Yes. Now, coming to further functions of ATP in cells. So, ATP is required in the cells for the active transport. There are two type of transport system. Yes, one is the active, another is the passive. Passive transport, if we are saying, means the from the higher concentration to the lower concentration, the content is moving. Yes. In the active transport, if we want the content to move from lower to the higher concentration, at that time we require the energy, we require the ATP. Yes, and that ATP is required to transport the content from lower to the higher concentration Yes. So, active transport means the hydrolysis of ATP provides the required energy for active transport mechanism to carry such molecules across the concentration gradient. Yes. At the time they are requiring energy. Then cell signaling. For many of the reactions which is occurring in the cell, the ATP is required. Many cascade reactions are activated through the ATP. Kinase is a very common enzyme which is requiring the ATP for generating the kinase cascade. Yes. ATP is also required in the DNA and RNA synthesis. Yes, the monomers are required in this the formation of this nucleotides. Now see other functions. So I told you ATP is required in cell signaling. Yes, intracellular, extracellular cell signaling. Many reactions or many enzymes are working with this ATP, with the help of this ATP. So it is also helpful in structural maintenance. The cytoskeletal elements are in their assembly just with the help of the ATP. Yes, ATP is also helpful for the muscle contraction. So ATP is binding with a myosin. That myosin is providing the energy, and that myosin will further facilitate to bind with the actin, that is causing the contraction and relaxation. Yes. Then there is a one topic called as a creatinine phosphate or creatine phosphate. What do you mean by creatinine or creatine phosphate or phosphocreatine? What are these terms? So see here, creatine phosphate is also known as phosphocreatine. This is a form of energy. Yes. Now it is an organic compound that provides a quick source of energy for the muscle fibers to contract. When there is a contraction in the muscles and we are doing the exercise, at the time this creatine is utilized, this creatine is providing the energy. Yes. Now this is also present in the brain, this is also present in the kidney, this is also present in the muscles. Yes. Now see the difference here, how the creatinine is formed. I am using two terms. See, one is the creatine and another is creatinine. Creatinine is the waste product and creatine is a form of energy. You can say they are formed from the sometimes from the amino acids like glycine or arginine, they are forming the creatine. This is a very common energy source. Yes. So creatine is synthesized in kidney, liver and pancreas. It is transported in the blood and it will go in the brain and muscle. So in brain and muscle it is stored. Yes, in the stored form. In the stored form it is living as a creatine phosphate, means with the phosphate molecule. Yes, and when there is a requirement of energy, this creatine phosphate will liberate the phosphate group and will give the energy. Yes. So here we can see in the brain and muscle it is remaining as phosphocreatine, and this phosphocreatine at the time of muscle contraction, whenever we are requiring energy, it will give the energy and then it will turn into the metabolic waste product, that is the creatinine, which need to be come out from the body. And if it is retained, this creatinine waste product is retained in the body, there will be problem. Yes. So we can see here there is a one chart, more chart. The creatine, which is the energy source, with the help of creatine kinase, it is in the phosphocreatine. Phosphocreatine is a stored form. Yes. Now this phosphocreatine can form the creatinine whenever there is a contraction, that can liberate the energy and creatinine is formed. Yes. Now this creatinine is a waste product, it is present in all body fluids, it will filtered out from the glomerulus or excreted in the urine. Yes. And the creatinine means the waste product, the value of this creatinine which is coming out in the coming out from the body. We can link the levels of this creatinine, means either we are taking a lot of proteins. Yes, we are taking a lot of means this non-vegetarian diet. Yes, that is increasing the creatinine in the urine. Yes, that will come out in the urine. Now, there is a one term BMR, basal metabolic rate. Now, basal metabolic rate is the number of calories. Yes, this is the number of calories which is required. Your body needs to accomplish the basic life sustaining functions, means at the time of rest. Yes, we are in the resting position, we are not doing anything, but at that time the calories are required. Why? Because there is some functioning is going in the body, like breathing is there, there is a circulation, nutrient processing is going in the body, the cell production is there. Yes. So, these are some of the common processes which is going in the body that is requiring some form of the energy, that is called as a basal metabolic rate. And this basal metabolic rate depends on some of the factors like sex, like weight, like height, age, ethnicity, weight history, body composition, some of the genetic factors. Like in the gender, if I will talk males, in the males the BMR is faster, basal metabolic rate is faster compared to the females. I will talk about the weight or height. Yes, the larger the body is, larger the BMR is. If I will talk about the age, in the old age the BMR, basal metabolic rate is slow. Yes, ethnicity, there are some races which are more they have the more BMR. Yes, weight history is there, of course they are, it is associated with weight, more the weight is, more the BMR is. Yes, body composition, how the body is designed. Yes, body composition is. Then there are some of the genetic disorders, there are some of the genetic factors which are associated with metabolic disorder, that can also affect the BMR, basal metabolic rate. I hope this is clear to all of you. Thank you so much everyone.